All right, hello. Let's start into chapter 5. We are really going to get into the nuts and bolts of how an antibody receptor or how a T cell receptor is going to be made. So let's go. All right. So, yeah, it's all going to be done by gene rearrangement. And it's actually the same mechanism that's going to apply to both antibody gene rearrangement and T cell receptor gene arrangement. And then once we have a nicely arranged front part of the antibody, like the part that's actually going to bind, we can then change the constant region. And uh, the antibody can have different possibilities. It can bind to different cells in different amounts, and it can work a little differently. So the immune system is going to try a lot of different things and is just going to try stuff, and whatever works is going to be reinforced. So when you get an successful antibody that actually can bind to the thing it's supposed to bind to, then there are going to be other cells that will give back signals that tell the immature B cell basically, well done, it looks like you got a winner here, and you can carry on with your development, and let's just see how far we can take this thing. That's the idea. So let's jump ahead. Okay, let's talk some of the vocabulary that we're going to do. There's a big section of vocabulary before we start to get into the pictures of things. And they are grouped, as always, by the section of the chapter they're coming from. So this should help you, if you haven't already read it, to figure out exactly what some of these things mean. And then go back, have a look at the chapter, because now we're into the complex stuff. But fortunately, it's all built on the things that we've been doing in Chapter 3 and Chapter 4. And that makes it a lot easier. Okay, first of all, we have variable regions. And so we know about those, right? These are the parts that are going to be different on every antibody or different on every T cell receptor, generally. And so, yeah, it's going to be the most variable part of the molecule, and it's usually, though not always, but usually going to be the antigen binding site because, I mean, why would an antigen would bind to all the antibodies if it bound at the non-variable sites. So, yeah, it wouldn't really matter which one you made, and you wouldn't be super specific potentially. So the heavy chain and the light chain both have their own variable regions. And if we're talking about a T cell receptor, then the T cell receptor alpha and beta chains each both have their own variable regions. And so this is very much the story of how we make and diversify and then modify, sort of refine variable regions on antibodies, immunoglobulins, or T cell receptors. Okay, within that V domain. So the V domain is an immunoglobulin-like fold. So think a whole bunch of little beta strands arranged in a particular way, one of those sort of squished beta barrels. And within a V domain, most of it is actually fairly standard and fairly constant, but there are 3 hypervariable regions where the sequence is just very different from one to another. And as you probably remember, these are called hypervariable region HV1, 2, and 3, which makes sense, right? Okay, and these are also sometimes called the complementarity determining regions, CDRs, because they're the thing that determines whether or not your antibody will bind to a particular antigen. So you'll see them, I think, most of the time in this chapter called CDR 1, 2, and 3, but it just means the variable part of the V domain, the variable domain. So the most variable part that determines specificity of an immunoglobulin or a T cell receptor. All right. Now, the way that we build these genes is, I think, the most interesting part of immunology. So within each of your cells, even cells that aren't part of the immune system, you have a library. And the library is going to have a set of V gene segments. It's actually going to have a leader segment that goes with each variable segment. And so, yeah, they're going to be roughly the first 95 amino acids of an immunoglobulin. Then you will sometimes have a D segment. And the D segment is, so the V is the variable, the D is diversity. And it's only little. It's about 5 amino acids, but they're right at the end of the V segment, and they can change the way the V segment, change the shape of it and the way it's going to interact. And so they can, yeah, be a little modification to the V. And then you get your J, which is the join segment. And so variable, diversity, join. And then at the end of that, you're going to have a constant segment, which is going to be the C. So VDJC is what you'll find in a heavy chain or a T cell receptor alpha, because those are the two big chains. If we're talking about a light chain, then it will only have variable, join, and constant regions of a light chain. And that's fine. All right. And so the way these things are brought together is we will actually, or rather the cells of the immune system will actually go in and edit their own genes. They will chop out and throw away large parts of their genome and select, we think at random, one of these gene segments. And it'll select one from each category if it's trying to make a heavy chain or a TCR alpha, or it'll select variable, join, and constant if it's trying to do a light chain or a T cell receptor beta. And this works because there are multiple copies of each segment. So there are many V's all lined up in a row, many D's, many J's. And so it's more a matter of the cell choosing the one that it wants, and then it just has to throw away everything else. And it has a wonderful protein that's going to help it do that. This is going to be a recombinase, and we will get to that a little bit later. So this whole process is called somatic DNA recombination. So we're talking about, this is regular cells of the body, in this case cells of the immune system. But these are actually going to do recombination. Now, recombination does normally take place during formation of sperm and egg cells during the meiosis stage. But we're not talking about recombination like that. We're actually talking about recombination that loses big chunks of the genome and would be very bad for making a little baby. You could build a little baby that would have exactly one antibody and exactly one T cell receptor if you did this recombination before the baby was born. And that would probably be great against some disease, but real bad against all the other ones. And yeah, you would just make a severely immunocompromised baby. And that's not what we want to do. That's not doing a good job. Okay. So the process that we're talking about is gene rearrangement. We're going to take these little segments, these little coding segments of the gene, and we are going to remove the parts in between that we don't want and remove all the extra copies that we don't want, and then join these little genes together, actually at the DNA level. Ordinarily, a gene gets joined together at the messenger RNA level. We start with a bunch of exons that are kind of spread out and have introns in between, and we will then join up all the exons and throw away all the introns. In gene rearrangement, we're actually going to do something like that process, but we're going to do it at the DNA level. And once a cell has gone through gene rearrangement, it cannot really go back because it will have thrown away all the other possibilities of things that it could become. So yeah, gene rearrangement can be permanent. All right, affinity means, strength of binding. That makes sense. Antibody repertoire means the total variety of things that your antibodies can bind to, or different types of antibodies that you can make. And so the total antibody repertoire of a human is somewhere between 30 and 60 million combinations of V, D, J, and C segments in the right order. But we don't just stop there, because if you only had to make, one from a library of 60 million antibodies, we actually have enough cells that are producing antibodies that you would always have all of your immunoglobulin repertoire produced at any one time. But what we do is we make those generic versions, and then we'll go in and specifically modify these things. So any antibodies that actually seem to be working a little bit will get encouraged to make further mutations. And these mutations can be most anywhere, but they can potentially make a better or worse version of the antibody. And so we'll do that and then select out the best ones and then send those off to fight whatever it is that's making us sick. Okay. The germline theory, exactly as it said, is we're talking about the theory that antibodies were encoded by a separate germline for each antibody. So this is the way that it works in us. And it is not true. So the idea was that there would be, instead of having VDJ and C regions that you could put together. The idea is that you would have just a series of antibody genes. You would have 60 million genes for entire antibodies, and you would then just run off a copy of whichever one you needed at the moment. So that was the first idea. It is not true um, for vertebrates like us, but things like sharks sometimes do have at least the V regions already pre-rearranged, so for ease of use, but for lack of diversity. Okay, the somatic diversification theories, this is the one that's a little bit more correct. So this is the idea that we have a small number of VDJ and C genes that are then diversified within the somatic cells. And so that's a way to take a little bit of genetic material and turn it into a lot of different antibody combinations, which sounds like a powerful way to do things to me, and sounds like a very good idea, and turns out to be pretty much what we do. So yeah, it's in contrast to the germline theory, which is not true for vertebrates like us. Okay, and then somatic hypermutation is the process that I mentioned at the end. So we're going to go in and put extra mutations into the V region of rearranged immunoglobulin genes, and we're going to produce variants of those genes that have probably never been seen before and may never be seen again. And this is the process of somatic cell hypermutation. We are deliberately mutating our own DNA after we have deliberately chopped out and discarded large parts of our own DNA. This is dangerous stuff. This is why the immune system is allowed to do it, and other cells are generally not. Although we will get into this a little later. There are diseases where these recombination enzymes, the ones that do the hypermutation or do the VDJC joining, where these are going to malfunction, and it usually leads to immunocompromise and some other problem on top of that. All right, so you remember your immunoglobulin fold? Yeah, I bet you do. Yes, two little beta sheets that are held together by a disulfide bond. Very nice, yeah. And variable gene segments are going to, have an immunoglobulin-like fold. And they're going to be, roughly the first 95 amino acids of the antibody or T cell receptor chain. And the diversity and join regions are the first ones that are going to get rearranged. And we'll show pictures of this to help you sort of get it in your memory. The way I think of it as D and J just go together. You got to have a DJ or else it's not a party. Yeah, so boom. First thing, you got to get a DJ. And then second thing, we're going to bring the V to the DJ. So we're going to recombine those things. We'll bring the V, D, and J together. And then we will swap in different constant regions as the antibody matures. And each one of those will have a different purpose. So the goal is to make one version of a B cell or a naive T cell with a pretty good V, D, and J region. One that happens to be pretty close to binding whatever it is that you are looking for, or that binds a little bit but maybe isn't as good as it could be. Then we make lots of copies, lots of clones of that cell, and the immune system will tinker around with each of those clones. It'll send them off to randomly mutate just some part of the variable segment, and in the hope that we will get a better antibody out at the end, or a better T cell receptor. Okay, here's a little picture. This is our immunoglobulin. This is the variable region shown at the top. There was an interesting paper that I just saw where they have, instead of actually making antibodies or nanobodies, like the little short-armed antibodies that you get in things like camels and alpacas, they actually just made a variable region from the heavy chain. So this VH region, just like is shown in panel A at the top. And they've figured out that this thing could actually bind to and neutralize SARS coronavirus 2. So I thought that was quite interesting. So we've got three nice hypervariable regions there. Those are the red loops. And in panel B, we've just kind of unfolded this protein so it's not like tangled up like If you would look at it in 3 dimensions, it's just sort of a schematic view. And the little black bar going over here, this is again the disulfide bond that's going to hold this together. So yeah, it's a couple of beta sheets that are stapled together with a disulfide bond. That's your typical immunoglobulin variable region fold. And if you were to look just at the gene itself, you can see that those hypervariable regions or complementarity determining regions are going to be spread out in roughly this order and roughly this arrangement. Okay, and so those parts, each of the little rectangles is going to be one of these strands. So this is strand A, B, C, C prime, C double prime, D, E, F, and G. The C prime and C double prime regions, I believe, are named that way because some animals have these and other ones don't. So I think a generic immunoglobulin fold is just going to be A, B, C, D, E, F, G. And I think in human immunoglobulins, we're going to have this extra copies of the C region, so C prime and C double prime there. And you can see that each one of those is going to have a little variable region in there. And so all this chunk at the beginning is going to form the V gene segment right here, and this little bit at the end is going to form the J gene segment. And you see that complementarity determining region number 3 is actually going to be in there twice. Or rather, you start out with, 2 segments, and either you're going to end up with this one or you're going to end up with this one. So you got 2 possibilities for that third complementarity determining region. And the diversity region would be right in between these, so in between the variable and the joining regions. You would have a D if it was a heavy chain or a T cell receptor alpha. Okay, so what are these diversity gene segments? Yeah, I need a little diversity in the workplace, right? A little diversity in our immune system. These are, very short little sequences. It's like a little tiny gene, or gene segment even, that is going to make a little join between V and J segments, and it's going to subtly, slightly change the way that they bind. And so you're going to find those in immunoglobulin heavy chains and in the, T cell receptor, I believe it's the alpha and beta chains, not the, or rather alpha and gamma chains, not the beta and delta chains. But they are saying beta and delta. So we're going to have to check that and come back to this lecture. I will check that and be back with the conclusion in the next segment. All right, so here it is in pictures, and this is showing you the process, and this is probably a good thing to commit to your memory, I would say. So you've got spread out on the genome various leader, variable, diversity, join, and constant regions. So we tend to leave the leader off because it's not actually going to code for part of the protein. It's going to make a little untranslated region before the variable region starts. This may be an example of evolution in progress. It may well be that eventually somebody develops a leader region that actually has a start code on, and so it starts the gene a little bit sooner, and so we would just add on then to the variable region and have an extra little chunk that could be switched around. So potentially that's where we're headed in the very distant future, and as maybe not as humans, but as vertebrate animals in general. So, leader, variable, diversity, join, and constant in a heavy chain, and just leader, variable, join, and constant in a light chain. And there we go. And the constant for the light chain, you'll see, is actually really short compared to the constant for the heavy chain. And the constant region for the heavy chain is actually what's going to form those that sort of handle of the antibody, the bottom of the antibody that can bind to cells. So this is going to form the FC, the constant fragment, if we were to break these with a protease like papain, for example. Okay, so some other terms that you're going to come in, and encounter in this chapter. Pseudogenes are things that look like genes, but they no longer encode a functional protein. So there's something wrong. Either They look like a complete gene, but they just don't ever get turned on. That can be a pseudogene. Or they can be a complete gene that's got a mutation somewhere near the beginning that means that it just doesn't get produced. It stops too soon or it doesn't ever start, something like that. So, for example, if you were to take something like birds, they actually have pseudogenes for some of the growth factors that you would need to produce teeth. And there are people that have done several experiments actually where you can take a little baby chick and figure out a way to turn these genes back on. These pseudogenes get turned back on, and you can make little tiny baby chickens that have toothy mouths, just like a little miniature dinosaur. Really cool stuff. We have a lot of pseudogenes in the human genome. For example, there's a gene that would normally synthesize vitamin C for us. The whole vitamin C thing is supposed to be part of a sort of a cyclic pathway that should go round and round. And in most animals, including things like whales, for example, it works exactly like that. And so whales don't have to eat vitamin C in order to survive. But in humans and human ancestors and a couple other groups, I think it's mostly primates and probably just primates related to us, the vitamin C synthase gene is actually broken. And so that's why we need to actually consume vitamin C, because instead of having a gene that would just do that for us, we have a pseudogene. So yeah, it's another little site if you wanted to genetically modify people in the future to make it easier to travel, for example, into space. Switching that thing back on, figuring out a way to do that would potentially have some real benefits. Yeah, it would mean one less nutritional requirement for those people. Okay, so anyway, back to antibodies. We have the kappa chain and the lambda chain. So there are two types of light chains that you can get on an immunoglobulin. You can select from the kappa group or you can select from the lambda group. They will both make functional immunoglobulin light chains, but each antibody can have only one. And so each antibody is either going to shut down or throw away all of its kappas and keep one lambda, or do the opposite, shut down or throw away all its lambdas and keep just the one kappa. And then a genetic locus just means somewhere on a chromosome, just a particular site on a long stretch of DNA. Okay. Class switching, that's a different thing. So that is when we actually change out the constant region, but keep the same variable and join or variable diversity and join regions. And I guess we would also keep the same leader region. This is something that B cells are going to do. So they're going to start out producing IgM, and then they're going to produce IgM and IgD for a little while. And then later on, they're going to switch, and they're going to stop producing IgM, and they're going to start producing either IgG or IgA or IgE. Yeah, that is class switching. That is how they do it. They keep the front end, and they don't throw away the other constant regions. They just hang on to them, and then when it's time, they can pull out the extra constant regions and yeah, keep the same variable region or FAB region really, and swap it onto a different base so they can bind to different cells and trigger different kinds of immunity. So here are the numbers, and if you wanted to multiply them out, yeah, you could multiply the minimum number here, so 34 times 5 times 1 plus 29 times 4 times 4, or you can multiply 38 times 5 times 1 and 33 times 5 times 5 and add those two together. So you've got to have a light chain, but it can be either a kappa light chain or a lambda light chain. And you've got to have a heavy chain, and so you can multiply these out. The low end will be 38 times 23 times 6 times 9. High end will be 46 times 23 times 6 times 9. And you can multiply heavy chains times light chains to get the total number of combinations, because you can have every single light chain in combination with every single heavy chain. And together you will find that you have in a human genome the capacity to produce somewhere between around 30 million at the low end, and I think it's 57 million, but we can say around 60 million different combinations of these genes on the high end. And this is just the start, remember. We haven't done our somatic cell hypermutation yet, but this shows you in numbers, how we get those numbers and how we get all these different possibilities. And the neat thing is, we can make 60 million genes, but we do it with only, look, you can add up all of these. You can add up roughly 34, 29, 38, 23, 5, 4, 6, 1, 4, and 9. You'd come to, I'm guessing, somewhere under 150. So let's say we got about 130, 140 genes, something like that. And together, these 140 or so genes are going to be able to make us 60 million combinations of genes. That's the power of the immune system, taking just a limited set of genetic material and turning it into a wonderful diversity of antibody types. And of course, we're not done yet, because we can take each of these types and add an unlimited number of random mutations into them through this process of somatic cell hypermutation. And so 60 million is the 60 million starting points. And from there, we basically go toward infinity in terms of the total number of antibodies that you could actually make. So there will be antibody combinations that you could make and that could potentially do you a lot of good. And you just may never get there because there are literally too many different antibody combinations out there to choose from. And a lot of parts of the process seem to be fairly random. So that's kind of the way it works. Okay. So here's the way an actual immunoglobulin gene looks. So for light chain lambdas or light chain kappas, you're going to have a bunch of genes organized like this. So for every variable region, so we'll call it variable and the lambda type number 1. It's going to have its own leader, and so we'll call that leader 1. And leader 1 is going to get attached to variable chain lambda number 1 whenever they get made. The leader's just not going to code for anything. It's not going to make part of the protein. It's going to make part of the messenger RNA that helps make part of the protein. Yeah, if that makes sense. And so you're going to have a series of these things, and you'll have, up to, let's say, 30 of these lambda variable chains. And then you're going to have a series of joins. And so you'll have join number 1 and constant number 1. Join number 2, constant number 2. But again, just in the lambda section. So just like that. And then you will have over here, and you see the little dots mean just there are more antibody chains in between. The kappa region is going to be organized in the same sort of way, except that instead of the join regions being split up in between the constant regions, we're going to have a little bank of joining regions in the kappa locus that allows us to actually go in and choose one of those without getting both a joining region and a constant region at the same time, for our money. Gives us a little more diversity. And then in the heavy chain locus, it basically looks like the kappa light chain locus in that all your join regions are going to be together, not separate. And in between, you're going to have a whole bunch of little diversity regions. It looks like up to 23 of those. And then they're just showing the first of the constant regions. And you see it's got a little Greek letter mu. So that means the constant region for IgM. And there will be a constant region gamma and a constant region delta for IgG and IgD, etc. Just like that. Okay, and I think that's a good place to pause this particular part of the lecture. We'll come back and plow on. There's really a lot here in Chapter 5, and it's worth taking our time and getting a real good idea of what everything is and what it does. So thanks very much.